The overall goal of the following experiment is to assess serotonin, or 5-HT, neurotransmitter function in a free-breathing animal with pharmacological magnetic resonance imaging and an intravenous challenge with the selective serotonin reuptake inhibitor fluoxetine. This is achieved by combining functional MRI with a drug challenge after initial baseline measures, which is used to visualize the brain hemodynamic response to the specific drug. Before the start of and during the experiment, extensive monitoring and maintenance of the animal's physiological responses is needed to ensure that the brain response visualized with fMRI is solely attributable to the pharmacological challenge given. Finally, statistical data analysis is performed on the MRI data in order to determine the brain's voxels, which exhibit signal variance compared to baseline values and are thus attributable to the serotonergic challenge. Results are obtained that give an indication about the brain's serotonin function based on the brain's response to a serotonergic challenge as measured with functional MRI.
The main advantage of this technique over existing imaging methods, like PET or SPECT, is that it is less invasive since no radio tracers are used. It is therefore a very suitable technique for translational research, as it can also be used in vulnerable human subjects, such as children or patients.
Though this method can provide insight into the functioning of the serotonin system, it can also be applied to other neurotransmitter systems when using a different drug challenge, such as the dopaminergic system, using, for example, an amphetamine challenge.
To begin this experiment, first fully anesthetize the rat, then cannulate the femoral artery and vein for blood gas and blood pressure measurements.